Kristina, first of all, can you tell us what was the main objective of your work? You focused on different subsets of unconventional T cells and their role as anti-tumor cells, as well as their immune modulation activities, protection against GVHD.
Thank you so much for the kind introduction. The work I conducted together with a group of people was focused on iNKT sublineages, and since a while we know that iNKT cells consist of at least 3 sublineages, and they are well characterized, but their functions haven't been clear so far. And so we investigated the in vitro and in vivo functions of the iNKT1, 2, and 17 cells. First, we came up with an isolation strategy to sort those cells based on surface markers. And then we performed RNA-seq and ATAC sequencing. ATAC sequencing is a method where you can actually detect open chromatin sites, and so can detect genes that are likely to be transcribed. And this has never been done on iNKT sublineages before. And we found in the RNA-seq and in the ATAC sequencing that the iNKT cells have the highest transcripts and also open chromatin sites for pro-inflammatory cytokines like granzyme B, perforin, and Fas ligand. And we barely detected any of those transcripts in NKT2 and NKT17 cells. On the other hand, performing the same analysis on NKT2 and 17, we found that they had the highest levels of IL-4 transcripts, or iNKT2 cells had the highest level of IL-4 transcripts, and also iNKT17 had detectable levels of IL-4 transcripts. So we generated 2 hypotheses. First one is that iNKT1 cells might be cytotoxic, might have cytotoxic potential, and iNKT2 cells and probably also iNKT17 might be more immunomodulatory. And to test these hypotheses, we performed, for the cytotoxic part of the project, we performed killing assay where we co-incubated tumor cells with isolated sublineages and performed FACS staining for dead tumor cells, and then later also performed an in vivo tumor model where we co-injected isolated iNKT sublineages together with tumor cells. And in this in vitro and in vivo assay, we found that iNKT1 cells had the highest cytotoxic potential, meaning that, for example, for the in vivo model, we found that the iNKT1 cells had a significant survival benefit, the mice that received iNKT1 cells, compared to mice that received NKT2 and NKT17 cells. And so this was for the cytotoxic part of the project. And then for the immunomodulatory part of the project, we performed a suppression assay where we co-incubated T cells with isolated sublineages, and then later performed the well-known major mismatch bone marrow transplantation model where we transferred 50,000 iNKT cells together with a graft. And we saw that NKT2 cells and also NKT17 cells led to a significant survival benefit, whereas mice that received NKT1 cells had no survival benefit and they died from GVHD. And then in an attempt to further investigate the mechanism of this GVHD protection, we performed RNA-seq on T cells after 7 days after transplantation, and we saw that the transcriptomic profile of these T cells was significantly altered when NKT2 or NKT17 cells were transferred. So, for example, we had lower levels of ITGAM, a gene that encodes CD49D, which is important for T cell migration to GVHD target organs. So to sum up, we showed for the first time that iNKT1 cells have the highest cytotoxic potential, whereas iNKT2 and 17 cells have more immunosuppressive immunomodulatory functions.
These are fascinating data since separating the graft versus host disease and graft versus tumor effect has been an objective for decades for investigators interested in allogeneic hematopoietic cell transplantation. So how relevant do you believe your data, your murine data, are to human transplantation, and how do you foresee developments using adoptive transfer of iNKT cell subsets in the future?
So, for your first point, I think our work, although murine work, is relevant to hematopoietic stem cell transplantation because GVHD remains one of the major risks after transplantation, and finding mechanisms to prevent from GVHD would be very useful. So, further investigations for example, of a transfer of immunosuppressive iNKT cells together with a graft, or the selective activation of these subset would be very, very interesting. And then on the other hand, transferring iNKT cells with a cytotoxic phenotype for cancer treatment would be, of course, another very interesting investigation to pursue. And then the second part of the question, cell-based therapies. I think iNKTs are a very suitable population for cell-based therapies because they have no graft versus host disease potential themselves, and so they would be also suited as off-the-shelf products or third-party products, which is a big advantage compared to conventional T cells. Considering the logistics we have nowadays for our cell-based therapies, being able to transfer off-the-shelf products would be a major improvement, I think.
Yes, thank you. And I recommend to listen to your full presentation again during the annual meeting. Read your paper in full and read the accompanying commentaries that was published in the same issue of the Blood Journal. To conclude this conversation on a somewhat more personal note, can you tell us what are your projects now that you are settled back in Europe and in Freiburg?
Yeah, so coming back from the postdoc at Stanford in Rob Negrin's lab, where I conducted the study together with Federico Simonetta and also Juliane Lohmeier, I moved on, of course, continuing my Hem-Onc fellowship at the Medical Center of Freiburg, currently conducting research also with Robert Seiser, and I just received my first grant to start my own group, and I will focus on this human iNKT cells and especially sublineages in humans, and also iNKT cell-based therapy in solid tumors.
Well, thank you for your answers. Congratulations again for your work, for receiving the Jon van Rood Award for this first grant. We wish you good luck, and EBMT is very happy that investigators such as you now join the community. Thank you again.
Thank you so much. It is an honor for me.